Hi class, this is Sir Alex Basco and welcome to our discussion in General Biology 2. Today we are going to discuss the second part of our lesson 6.2, which is the light-dependent reactions. This is still part of our discussion in photosynthesis, and light-dependent reactions is actually the first half of photosynthesis. So last time we've discussed Hi class, this is Sir Alex Basco and welcome to our lesson in General Biology 2. This time we are going to discuss the second part of our lesson 6.2, light-dependent reactions. So in lesson 6.2, first part, we've discussed photosynthesis. We've got a little bit of our introduction in photosynthesis, and we learned that there are actually two steps of photosynthesis. The first one being the light-dependent reaction, and the second part being the Calvin cycle, or the light-independent reactions. So for the second part of our 6.2, we are going to discuss in depth what happens during the light-dependent reactions. So light-dependent reactions is basically the part of photosynthesis where we use light energy to make ATPs and NADPHs or NADPHs. So meaning the end goal of our light-dependent reactions, or the first part of photosynthesis, is to convert light energy to chemical energy in form of ATPs and NADPHs. This is important because in order for us to continue on the second part of our photosynthesis, called the Calvin cycle, we need to create ATPs and NADPHs. ATPs are energy carrying molecule, while NADPHs are actually electron carrying molecule, and we need this in order for us to undergo Calvin cycle. One important thing to remember about the light-dependent reactions is that it happens in the chloroplast, and more specifically, if you remember the site of photosynthesis, this light-dependent reactions is happening on the thylakoid. Thylakoid are the disc-shaped structure that you will find on your grana, and on those thylakoids, that's where you will actually find your chlorophyll or the green pigments, and the green pigments are the one necessary in absorption of the light. So speaking of absorption, absorbing light or using of the energy from light, there is a system that is specialized in harvesting this light, and that is called the photosystem or photosystems. And photosystems are basically large complexes that are made up of proteins and the pigments. So the pigments you will find, although we say it's chlorophyll, there are actually various types of chlorophyll. You have your chlorophyll A, chlorophyll B, C, D, and carotenoids. So all of these various pigments along with proteins are bunched up together to create complexes called the photosystems. And these photosystems on your thylakoid are necessary because first, they harvest the light, and second, they played a major role in the conversion of light energy to our ATPs and NADPHs. There are two types of photosystems that we will encounter today, photosystem 1 and photosystem 2. The whole process of light-dependent reactions, you would notice that we will always start on photosystem 2. Although this is numbered 2 and this is numbered 1, and the step-by-step process, we start on photosystem 2. The reason that we just call it photosystem 2 is that it's discovered after photosystem 1. So photosystem 1 was just discovered first, hence we call it the number 1. Photosystem 2 was discovered afterwards, hence we call it number 2. But in the whole process of light-dependent reactions, we will begin with photosystem 2. So where do we find this? Basically, the light-dependent reactions, we also call it the non-cyclic photophosphorylation. Phosphorylation because we are creating ATPs. I hope you guys remember your ATPs. There is an energy usage. So we added photo because this is a light-driven phosphorylation, and we call it non-cyclic because it's linear. So the standard process of light-dependent reaction is actually linear. There's another type of light-dependent reactions which is cyclic, and we will discuss that later on, almost at the end of our lesson. But let's look at the general process of what is happening in the light-dependent reactions. So like I've mentioned, this is all happening on the chloroplast, more specifically on your thylakoid. And on your thylakoid, that's where you will find your pigment, and more specifically, those pigments are found on your photosystems, and they are bunched up together alongside proteins. And these photosystems, like here you go, photosystem 2 and photosystem 1, are the one necessary in harvesting or absorption of light. So as you can see here, you have your light energy coming from the sun, and they are absorbed by your photosystem. And eventually what happens is that the photosystem, called the reaction center, are the one necessary that are the ones that are actually donating your electron. And as you can see here, if you notice a little bit of similarity with your cellular respiration, this is just a whole process of electron transfer. So the electron that comes from your photosystems are being passed on to various molecules, and what happens is that as this pass on, you are creating your proton gradient, similar with what we do in our cellular respiration. And this proton gradient is the one necessary to activate your ATP synthase in order for you to create ATPs. And also, as your electron gets getting transferred, eventually there would be an enzyme. There's an enzyme right here that will convert the NADP plus and add that electron to create your NADPH or NADPHS. So eventually these ATPs and NADPHS will then be used on your Calvin cycle. So this is we call it non-cyclic, or basically it's a linear process because it's just straight forward. The electron does not go back creating a loop, but they're just going in a straight manner. But again, like I've mentioned, there is a cyclic process that we will discuss later. So before we go in depth on what is happening on each of these individual processes, let's go and take a look at the basic steps of non-cyclic photophosphorylation. So the first one is the light absorption in PS2 or your photosystem 2. So what happens is that light gets absorbed by your photosystem 2, more specifically by the pigments on the photosystem 2. And when light is absorbed, they are excited, meaning their energy levels becomes increased. And what happens is that these pigments actually transfer this energy from one pigment to another. So you can see here, you can think of this disk right here as your pigment, and the energy is being bounced from one pigment to another until it arrives at what we call a reaction center. This reaction center of your photosystem 2 is the one that will donate your electron. So it will donate that electron, and that electron will be the one used for your electron transport chain. When that reaction center donates electrons, of course, it removes electrons. So it's missing an electron. So what happens is that it has to replenish or refill that electron. And it gets that electron from splitting water. We usually have to split H2O. And that's where we will get our electron to replenish the missing electron from the reaction center. So I'm gonna do this now, but later on you will see this again. So how do we split electrons? When we talk about splitting electrons, we are not or splitting water in our first part of our light dependent reactions. We're not talking about just splitting H and O. Okay, we're not only talking about splitting our H and O. What we're actually splitting is the H. Remember, H is just a atom, and this atom has one proton and one electron. This whole H is basically a combination of one electron and one proton. So what happens is that when we split these two, we remove the oxygen. We're actually splitting two molecules of water. So we ended up having two oxygen, and this oxygen is released via the stomata, and that's the one we breathe. And then our H is being split with its proton component and its electron component. So what happens in the end is we ended up having 4 protons and our 4 electrons that is being transferred. These 4 electrons then will replenish the electron missing from your reaction center, and these 4 protons, which we usually also write as H plus, is the one that will be added onto your proton gradient. I hope that is clear, but we will discuss this again later. So again, so the reaction center needs its electron replenished because it donates its electron after it receives energy from the sun as the pigment transfers that energy. So you have to split your H2O. So if you remember the light dependent reactions in our discussion in 6.2 part 1, H2O is being used on light dependent reactions because we have to get that electron to replenish our reaction center, and the 4 protons, this H plus right here, is the one that will create your proton gradient. So what happens then is this proton gradient is used to create your ATP synthesis or ATP synthase, similar with how cellular respiration happens. Then the third part of our light dependent reaction is that the light absorption in PS1 or the photosystem 2. So remember, so after our reaction center from PS2 donates its electron, it will move to a primary, it's an electron acceptor, blah blah blah. It will go to our electron transfer. We will go in detail how this happens until this H or this electron plus would actually ended up in photosystem 1. The reason for that is because in photosystem 1 there's another reaction center that donates its electron. So in photosystem 1, you have another reaction center right there that donates its electron. So when it donates its electron, it has to replenish its electron, right? The electron that it needs to be that is used to replenish the missing electron is the one that is coming from PS2. So when the electron is being transferred, so this is your electron transfer now, it will go to PS1 to replenish that missing electron. And then this electron that comes up from PS1 is again used on another electron transfer to pump H plus, but also to finally create your NADPH system, because this electron will eventually end up with a what we call a PC. It's an enzyme that will convert your ADPH or NADPH to NADPH, NADP plus and H to NADPH. So this electron will be the one necessary to create your NADPH. So I hope the whole process for now is clear. That is just the general gist of what is happening. Now what we're going to do is we're going to go in depth with the whole process. Okay, now let me remove this. Okay, so one thing, but before we go in that, one thing that I want you guys to remember is ultimately what is happening is there is an excitation of P680 and P70. This P680 is the reaction center of PS2. Okay, they're actually special. They're actually special, what we also call special pairs, because they are actually a pair of chlorophyll. Okay, 680 number comes from the wavelength of light they absorb. The wavelength of light they absorb is about 680 nanometers of wavelength. Hence, that's why we call them P680. So when the energy from the sun is transferred to the pigment, and the pigment will transfer this or funnel it out to their reaction center, the special pair 680 or P680. This P680 will become excited, and when it becomes excited, it will readily donate its electron. So the energy from its electron increases, and then when it increases, it can easily donate that electron, and then it will go on to that whole transfer process until it reach the PS1. The PS1 here has another special pair This is another name for P700 special pair, or the name of the reaction center, P700. This P700 also, when the light energy from the, so you have your sun, it gives light to the pigments found on your PS1, and that pigment funnels that energy to the reaction center called P700, which is another special pair of chlorophyll, and this becomes excited again, and when it becomes excited, it readily donates its electrons. So when it becomes, there's an excitation happening here. And when it's excited, it loses electron, so the electron here is the one that will replenish the missing electron there. And the missing electron here that it just donated comes from splitting H2O. So again, that is the whole process. I'm repeating this because it can get confusing, but I hope so far is clear. So if there's any questions so far with this, I hope you guys read your module. It will help you understand what is happening. Okay, so now let's go in depth with our discussion. What is a photosystem? Again, photosystem are a combination of photosynthetic pigments such as your chlorophyll A, chlorophyll B, and your carotenoids, along with proteins, and they are light harvesting complexes. When a pigment absorbs a photon, or photon is the light coming from the sun, it becomes excited, which means that one of its electrons is boosted to a higher energy orbital. So when something gets excited, the electron gets excited, what happens, your pigment right here, so it receives photon. So this is similarly what's just discussed earlier, the picture that I showed. So it becomes excited, right? That electron right there. When we talk about getting excited, the electron moves from the next orbital. Okay, so this is your atom, this is your photon right here, or your proton, and usually it has an electron on its s orbital. Remember, this is the first orbital where it can contain 2 electrons, and when it gets excited, some of those electrons moves to the next orbital or to the next shell. That is basically what is happening, and when that happens, this whole process is unstable and it needs to donate that energy, or well, in this case for pigments, it has to donate its energy in order for it to become stable again until it reaches the reaction, or the reaction center, wherein instead of donating the energy, it will instead donate the electron, which will initiate the whole electron transfer chain. So most of the pigments acts as an energy funnel, transferring their energy to their neighbor pigments via resonance electron transfer. So like I mentioned, the only pigment, or the only chlorophyll pigments that transfer its electron is the reaction center, or your P680 for PS1, or your P700 for PS2. The rest of the pigments, when they become excited, when they receive photon, or when they receive light energy, they transfer the energy itself, not their electrons, and they transfer that energy via resonance electron transfer, and until such a time that it's basically so it's right here, these are your pigments right here. So instead of donating that electron, it transfers the energy itself from one pigment to another pigment until it reaches the reaction center. Again, this reaction center is a chlorophyll special pair. It's a special pair of chlorophyll A, and when they receive that energy after it's getting transferred from one pigment to another, so the pigments funnel that energy, it will donate its electron to what we call a primary electron transfer. So for primary electron transfer for PS1, there's a special name for this primary electron transfer, but again, so it will donate its electron, and after donating its electron, it will donate it first to the primary electron acceptor, and then this will start the whole electron transport chain. Okay, so this whole process is very similar with what's happening to both PS1 and PS2. So let's distinguish a little bit more the difference between PS1 and PS2. So again, there are two photosystems, PS1 and PS2. The main key difference is that for special pairs, PS2's special pair absorbs 680 nanometers of wavelength. That's why we call it P680. PS1, a special pair, absorbs 700 nanometer of wavelength. That's why we call it 700 or P700. Another difference is their primary acceptor. PS2's primary acceptor first passed their election. The reaction center of PS2 first passed their election to what we call a phaeophytene, which is the primary acceptor, and the phaeophytene will transfer this to initiate the electron transport chain, while the PSI or the PS1 photosystem 1's reaction center donates its electron to the primary acceptor called A sub naught or A naught. And lastly, the electron source. So again, when the reaction centers from PS2 and PS1 donates their electron, they're basically missing an electron. PS2 replenish the electron from splitting H2O. Again, remember, when we split H2O, not only we are splitting it with H2 and O2, but we are also splitting the H into its electron component and to its proton component. That's where you have your H plus right there. The electron component is used to replenish your PS2's reaction center, while the proton is added onto the proton gradient. While for PS1, replenish its electron from basically the electron that the PS2 donated to its phaeophytene onto its transport chain. Okay, so let's go to photosystem 2. Again, photosystem 2 absorbs energy that has a 680 wavelength, and when it gets excited, it donates its electron to phaeophytene, which initiates the electron transport chain. After the P680 donates their electron, they become positive, so new electron is provided from splitting H2O. H2O does not easily donate these electrons, hence we have what we call a manganese center. The manganese center is actually the one that has to split your H2O, and two molecules of H2O is split, creating 4 H plus, so you are actually creating 4 H plus and 1 molecule of O2. Okay, I hope that is clear so far. I've just been repeating the whole process. So this is what's happening on the photosystem 2. After that, so the whole process has finally started. Now let's go to the electron transport chain and to the PS1. So the electron transport chain and photosystem 1. So the electron from photosystem 2 moves to what we call, so after it moves, this is your phaeophytene or your primary meaning the first one to accept the electron from the special pair or your reaction center. So it receives that electron, and this phaeophytene will first donate the electron to what we call a protein called plastoquinone or PQ. Then from plastoquinone, it will move to cytochrome complex or CYT, and then from the cytochrome complex, it will move to plastocyanin. Okay, so as you can see there, so the E transferred to plastoquinone to cytochrome complex to plastocyanin. And plastoquinone and cytochrome complex are actually, when they receive these electron, one interesting thing is that the electron first it's getting excited. So this is you can think of this as the energy level. And as it gets transferred, the energy level of your electron actually decreases as it gets passed on to this molecule. And the reason for that is because these molecules right here is using that energy from electron to pump the H plus. So it's pumping a proton from the this is stroma. Stroma is the outside fluid filled structure of your chloroplast, and this is basically your thylakoid right here, and this is the inside of the thylakoid. That's why we call it the lumen of the thylakoid. So it pumps proton from the stroma to the inside of your thylakoid. Okay, and then the electron moves from higher to lower energy. So the electron, as they move from higher energy to lower energy, because as energy is released, this plastoquinone and cytochrome C uses that energy to pump H plus to the interior of the thylakoid. And then eventually, when there's so much H plus inside the lumen of the thylakoid, this H plus then moves downhill. So it moves from area, basically just simple osmosis, it moves from an area of higher concentration to an area of lower concentration. Remember our discussion on osmosis? So since we have so much H plus here, they will just normally would have to move when there's a place for them to move the H plus, and the molecule necessary for the H plus to move back to the stroma is basically, this is again your stroma right here. There's a lower concentration of H plus here now because we keep pumping those H plus inside the thylakoid. So there's higher concentration of H plus here, and there's lower concentration of H plus here. So the H plus moves from lower concentration or from higher concentration to lower concentration. Basically, they move downhill, and when they move downhill, they use the ATP synthase, and when they move downhill, there's energy there which converts our ADP and our phosphate to ATPs. That's how we create ATPs. And then lastly, after the plasticyanine will transfer this electron to the reaction center of PS1 or our P700. So the electron arrives to PS1 and joins the P700, because the P700 is again missing their electron because it donated that electron, because the pigment, as you can see here, it's similar with our discussion earlier of PS2. So the pigments in PS1 are invigorated by the light energy, and that light energy is transferred from pigment to pigment until they arrive at your P700. And when this gets excited, it again donates its electron. So now it's missing electron, so the electron from plastocyanine is transferred to your P700. So what happens is that this P700 first donates its electron to your A sub naught, which is your primary electron acceptor, and then from there the P700 donates the electron to A sub naught, then to what we call a ferredoxin or our FD. And then lastly, the ferredoxin will donate this electron to an enzyme called the NADP plus reductase. This enzyme then uses that electron and create your NADPH. Yes. So this NADP reductase will create your NADPH after they receive that electron. So now we have our NADPH and our ATP, and I hope it's clear how the whole process of the first part of photosynthesis is happening. So this is the non-cyclic photophosphorylation, or basically your linear photophosphorylation. Because if you look at this, there's an arrow going here, right? So this arrow here is creating a loop. So sometimes the ferredoxin, instead of donating that electron to NADPH, it sometimes return that electron to either plastoquinone or to cytochrome C, creating a loop. And that loop is now what we call a cyclic electron flow. So cyclic, so the linear photophosphorylation, or the non-linear or non-cyclic, photophosphorylation is the standard process where we create ATP and NADPH But there's again another process which breaks this cycle called the cyclic photophosphorylation, wherein the ferredoxin moves the electron not on your NADP reductase but moves it back to your plastoquinone or cytochrome C. And the important thing about the difference between the two is that the cyclic photophosphorylation drives the creation of more H plus gradient. So it is necessary, if the cell needs more H plus gradient, it will undergo the cyclic photophosphorylation. If not, it will undergo linear photophosphorylation. There are many factors that determines whether which photophosphorylation the cell will do, or this plant cell will undergo too. In some cases, they can switch from linear to cyclic. So for example, if there's too little NADP plus, so if you don't have enough NADP plus to create NADPH, what it will do is, instead of donating that electron, it will just return it back. And some plants that is specialized more in creation of ATP, they would rather prefer they would rather also prefer using the cyclic, because it will create more H plus gradient to create your ATPs. And lastly, prevention of damaging the photosystem from excess light energy. So if there's too much light energy, there's too much electron happening, but again, probably not enough NADPH. So instead of throwing away that electron, it will keep returning it, creating this whole cyclic process that will keep pumping the H plus. So that is necessary if there's too much energy from the sun. So in order for the plant to exhaust itself or to damage its photosynthetic photosystem, it will do that. Okay, so I hope it's clear now what is happening on our light dependent reaction. If there's anything that confuses you, please do not hesitate to message me. Thank you so much, guys, for your time, and this is the end of our discussion. Our next topic will be about Calvin cycle. Thank you so much, and may you guys have a great day.